From the Biological Psychiatry family of journals, this is the Biological Psychiatry Podcast, where we discuss the latest research on psychiatric disorders and their treatments. Thank you for joining us. I'm your host and social media editor, Elvisha Dhamala. Clinical trials are research studies involving human volunteers designed to evaluate the safety and effectiveness of new medical treatments, drugs, devices, or diagnostic tools. These trials have multiple phases. Phase 1 focuses on safety and dosage with a small group of people, typically healthy volunteers. Phase 2 evaluates efficacy and side effects in a larger patient group. Phase 3 confirms efficacy and monitors side effects in large-scale, often randomized international trials. Phase 4 occurs after FDA approval to monitor long-term effectiveness and safety in the general population. Clinical trials are very expensive and prone to failure. In fact, psychiatric clinical trials face some of the lowest success rates among major disease areas. Today, we're exploring a novel approach to improve these odds, one that uses something we all do in every conversation: the way we speak. Joining me, we have Dr. Alex Cohen from Louisiana State University, first author, and Dr. Mark Opler from Clario, co-author of a recent paper in Biological Psychiatry that demonstrates how analyzing speech patterns can improve clinical trial outcomes. Dr. Cohen and Dr. Opler, welcome to the Biological Psychiatry Podcast.
Thanks for having us.
Thank you.
Psychiatric trials fail at much higher rates than trials for other diseases. What makes them so challenging compared to trials for other diseases?
There's a lot of reasons that trials fail. I think the principal concern in clinical trials in psychiatry is our inability to differentiate drug effects from placebo. And this is partially due, I think, to the inherent noise in any experiment, in any clinical trial. But in psychiatry, we have the added problems of, first, highly subjective endpoints. We're often asking people about their subjective sense of improvement. We're evaluating sometimes relatively subtle changes, and again, subjective changes in behavior. But we're also trying to study diseases whose cause and underlying biology we simply don't fully understand. We're diagnosing on the basis really of syndromes. Unlike, let's say, a study where we're looking at blood pressure, where we strap a cuff on somebody and numerically measure how much higher their blood pressure is today than yesterday, we don't have a cuff that we can strap on you to measure how depressed you feel or how much better or worse your hallucinations are today than yesterday. So I think the subjectivity combined with other sources of noise, both in the diagnosis and the evaluation of these diseases, creates a tremendous challenge. And when we're trying to conduct science, the artistic aspects, subjective aspects of this often make it really challenging. This is why I believe that the move from subjective to more objective endpoints that are closer to the underlying biology of whatever these diseases actually are is absolutely vital.
Yeah, this is an essential move towards precision psychiatry, is the use of some objective biomarkers that are closely tied to the psychopathology, to functioning, and to the underlying biology.
So in this paper, you mention enrichment strategies. Can you explain what these strategies are in the context of clinical trials and what makes them so important?
Enrichment strategies are using patient characteristics to select participants that somehow enhance the signal detection of drug effects. These are done prospectively, so before randomization. And there's 3 general strategies, but they involve things like trying to reduce placebo effects, or identify people that are more likely to show placebo or very quick responses that are unlikely due to drug effects. Strategies for identifying high-risk patients that unambiguously show the disease state, or identifying patients that are more likely to be responsive to treatment.
So that's a great segue into the next question. You mentioned earlier for something like blood pressure, we have a blood pressure cuff. So again, you needed something more objective in psychiatry, and you decided to focus on speech. Why did you focus on speech specifically, and is there something unique about how we speak that makes it important in psychiatry?
Speech is a critical human ability, right? It's important for most people as they navigate their daily routine, navigate relationships, work, recreation. During a clinical interview, it's the primary media that's being, evaluated. And it's something easy to objectify. Speech technologies are well established. They've existed for at least 70 years. They've increasingly become sophisticated and more scalable. And I think their promise for capturing speech, not just for a few languages, but for people all across the world, is improving very quickly. Within the clinical interview, speech biomarkers can be extracted, from interviews that are already being conducted. So there's no additional burden on the participant or the study team, no additional blood draws or tests or interviews.
So you looked at speech latency in particular. Can you tell us what that is and why it's important to measure how someone is pausing before responding?
Yeah, of course. Speech latency refers to pauses. And here we're interested in one particular type of speech latency, which is a turn latency. So that's the time in milliseconds between when one speaker stops talking and the other speaker starts talking. So the interview asks a question, "How are you doing today?" And the patient processes the question, formulates a response, and then begins articulating the response. And turn latency is the time that it takes for those mental processes to occur. The psychiatric interview or the clinical interview is ideal for extracting speech latencies because it's structured, it's typically delivered by trained personnel. The questions are challenging, so it puts a bit of a processing load on the patient. There are follow-ups, and there are also lots of turns in an interview, so you get a reliable estimate.
You ultimately ended up using speech latency ratios rather than just the raw latency itself. Can you explain what that ratio captures and why it works better than just response time?
We did some work with depression, and we saw that speech latencies were a good measure of psychomotor slowing. And we think with depression, what happens is people may be slowed. It takes time, extra time to process and articulate a meaningful response, but they're able to articulate a meaningful response. I think when patients have more profound social cognitive deficits, the response is also affected. And the speech latency ratio accounts for this. So we're including the latency, which is a measure of psychomotor slowing, but also the duration, which is a measure of response. And together, we think that captures a person's ability to engage with the interviewer more than just their psychomotor response time. There's no doubt that duration is a kind of crude measure of somebody's response, right? It doesn't include the sophistication or the number of concepts that people are proposing, or even how linear the response is or how good of a response it is to the person's question. And there are many, many creative and sophisticated ways to try and quantify somebody's response, particularly using LLMs. But our use case here required our measure to generalize across languages and across very different people. And there are big challenges in using LLMs that way. So duration seemed like a simple and interpretable and transparent way to capture people's response.
One thing that is really impressive that you just mentioned is this was a global trial with 8 different languages. So, how did you ensure that any biomarker you find would actually be generalizable and would work across such diverse populations?
So we first consulted experts in the languages when they were available. We put in place a quality assurance pipeline to ensure that the tech was working appropriately across these different languages because they use different modeling and different training techniques. And then we examined the reliability and validity of our ratios across the languages. It was a bit of a challenge in that the sample size varied across these languages. But generally, we saw that the ratio performed. The mean values sometimes were different across languages, as would be expected, but it seemed to perform psychometrically well across all languages.
So, the study that you did was building off of a phase 3 trial for a schizophrenia drug. Can you describe the study design of the clinical trial itself and how you incorporated your speech analysis into it?
The design of the study was very similar to many pivotal trials in schizophrenia for new treatments, where you have randomized into one of two arms, patients who are either going to be put on to placebo matched for all the characteristics that we believe matter, right? Appearance, the pill looks exactly the same, that sort of thing, or on to one of two doses of active treatment, a low dose and a high dose. And again, this is fairly standard, I think, across almost every large trial that we see. Obviously, the details will differ. But the idea here was that we were not changing anything about the underlying trial. In fact, the underlying trial had already been conducted, so there was no way for our approach to affect the results. For this study, we had several advantages. I think one is there was already employed a set of technologies that enabled us to very easily and effortlessly capture the audio. So this study, instead of using paper forms where clinicians had, their interviews and all of their questionnaires, there was actually a tablet that was provided to every site by Clario, so that every assessment, everything from the clinical global impression scale to the positive negative syndrome scale or PANS, scales to assess motor function, all of these things are being collected electronically. And the PANS assessment, which was the primary outcome, PANS being the gold standard for assessment of efficacy of treatments in schizophrenia, this was captured electronically along with video recordings of each of those assessments in every language for every patient everywhere in the world. So that was the essential design. One of the beauties of this approach is that it's invisible. You don't really see it. Whether you're recording audio on a tablet or through some other means, it's become such a standard part of psychiatric evaluation. Nobody really sees it anymore.
So, is it safe to say that speech is perhaps a far more accessible biomarker than potentially some of the other technologies that are being considered for biomarker development?
I don't think there's any question about that. Speech is one of the things that defines our species, isn't it? It's what differentiates us, I think, from other primates. And I would add to that, speech is probably one of the most complex behaviors, one of the last to emerge neurodevelopmentally. Because of this, I believe it utilizes so many resources that we have to engage as human beings. Social resources when we're interacting with another person, cognitive resources to dredge up, process, and formulate the information, neuromotor resources to deliver it. All of this comes together in speech and in the act of a clinical interview, which again is a very standard part of any clinical assessment, especially in psychiatry. It's the most ubiquitous and probably the most accessible form of what I would regard as biological information we have available to us. And what makes speech latency and speech latency ratio even more beautiful, in my view, is the fact that it generalizes across every language, every culture, every country, every person we've evaluated using this technique. This is valid, this is replicable, and it's meaningful.
So, in the study, you analyzed over 2,300 recordings from psychiatric interviews. Can you talk a little bit about your main findings? So, when you enriched the clinical trial sample, what did you see in terms of the treatment effects?
Yeah, the treatment effects improved, typically dramatically, sometimes doubled in nearly every primary and secondary endpoint, total and subscale score. We saw statistically significant improvements from baseline to end of treatment in 9 out of the 12 outcome measures. And this is impressive because we had half the participants. So a big power reduction there. I think this was particularly impressive for negative symptoms, where the treatment placebo separation jumped from about 0.39 to 0.71 with the enriched sample.
Let's talk a little bit more about that. Can you tell us why negative symptoms in particular are so important in schizophrenia trials and why perhaps they're so challenging to address?
So negative symptoms have been a target for treatment for a long time. They're one of the things that we've been struggling to both understand better and also to treat more effectively. Mostly because there are good reasons to believe that this is the primary barrier between patients and their recoveries, the resumption of the lives that they lost before they became ill. is really blocked by these deficits in motivation, language, engagement, interest. There are lots of good reasons why, the world wants to find a better treatment for negative symptoms. The study that we're talking about today was actually not a negative symptoms trial. It was a general trial for overall antipsychotic efficacy. Yet, negative symptoms proved to be incredibly important in defining a group of patients who really had meaningful responses to drug. I believe this is because, even when our focus is on the more obvious, the more classical symptoms of psychosis, negative symptoms are still a fundamental part of that disease and disease process. And I think what speech latency ratio does, it identifies patients who have these fundamental deficits and who probably have a very specific form or phenotype of the disease that is particularly sensitive and responsive to drug treatments.
Interpreting ratios is very much still in its infancy. And so with this study, we were primarily replicating and extending a study of bipolar depression that we'd done with Seth Hopkins, with Steve Szabo, and Josh Siegel, who were then at Sumitomo Pharma. And the process requires some symptoms to anchor our speech biomarker in, so we can decide what a cutoff value should be for enriching the sample. So we decided to use negative symptoms for this study, and that was primarily for, faithfully replicating our methods. Since then, we've moved on, and now we're looking directly at placebo effects and treatment response across a wide variety of symptoms, including negative symptoms, but also, general psychosis, mood and anxiety, and things like that.
The effect sizes that you mentioned are very impressive. So you could nearly double the drug placebo effect with half the amount of participants. What does that actually mean for clinical trial design?
To me, the most important extension of that effect, I think, is the reduction in the size, the complexity, the length, and the cost of clinical trials. I also think that, ethically we have to think about the implications too. A lot of people ask, why are clinical trials now so difficult to do? 20 years ago, 30 years ago, we could do studies that were much smaller, much more effective, and they didn't take as long. Now it feels as though there's an entire industry that is ballooning and the costs are just spiraling out of control. What I see within the scientific elements of the work that's been done, I see a promise of smaller, more efficient, less costly, and more ethical trials where we identify a specific group of patients with a well-defined phenotype who actually stand to benefit from the treatment that we are investigating. Costs aside, timelines aside, that to me is worth all of the effort that we've expended. And I look forward to seeing a day when we can do faster, smaller, more precise trials.
Getting back to speech latency, what do abnormally long speech latencies tell us, and what cognitive or social processes might these be tapping into?
That's a really good question. And I think that's one that we're still disentangling. But I think it's clear that during an interview, at least, this measure of ratio is an indicator of somebody's engagement in the process. An engagement can reflect many factors, some cultural, some contextual, but certainly cognitive, social, and motivational factors, and ones that are important for psychopathology, especially serious mental illness like schizophrenia and psychosis. We sort of engineered this measure of ratio so it would be relatively stable over time, and it would be used during the baseline and screening sessions of a clinical trial. So I think what we were really interested in is identifying people who were disengaged, people who were really struggling to engage during the session. And this abnormally long ratio, which is caused by long latencies and short response durations, I think very clearly reflects somebody who's not engaged in the session.
In your prior work, you also found that there was a speech latency biomarker that's useful for enrichment in a phase 2 antidepressant trial. What's similar about that work versus this work, and how might the work that you're doing overall generalize across psychiatric illnesses in clinical trials more broadly?
There's a lot of similarities. The methods we used are almost identical, particularly for making decisions around enrichment. The general results very similar, with about half the subjects and in some cases a doubling of the effect. We used the same automated speech analysis pipeline, and the features seem to work across sociodemographically diverse people. And in particular, in that study, 7 languages. I think the differences are simply the feature in that we were looking for latencies and speech latency ratios in this study, the drug and the population. The reason why it's working may be slightly different. In the prior work, what we saw was fast speech latencies identified people that were primarily showing a placebo response. In this study, we do see some placebo response, but we see also an enhanced treatment effect in people that had long ratios. So we still have lots of work to do in terms of figuring out exactly what's going on functionally and neurobiologically, but
So, what would you need to see before this becomes standard practice in all clinical trials?
I feel that for certain applications, for certain use cases of this approach, the data exists today to consider, the validity and the reliability or the reproducibility of the effect. I think it's also worth pointing out that right now there are several large studies ongoing that are using this prospectively, meaning they're actually evaluating speech latency and speech latency ratio as patients are being screened into the trial. And that data is being used in a variety of ways. So, the case for the approach, I think, both in the recent publication and in the prior publication, are very well made. I do believe that we need to complete analyses of additional data sets and also to really dig into the underlying issues, right? So why, for example, are certain patients classified one way and others another? What's different about these two groups? In depression, we're identifying people who may have depression, but maybe not the biologically mediated depression that really requires drug treatment. That may differentiate the vocal biomarker negative versus positive, the slow versus the fast speech latencies. In schizophrenia, or the schizophrenias, depending on your point of view, it will probably wind up being more complicated than that. But again, I suspect that the overall shape that it takes as this method progresses will feel very much the same. I don't even necessarily know that this is all that specific to depression or to psychosis. Uh, you know, Alex has also done some fascinating work in neurodegeneration that suggests a role for speech latency. And I think we have good reason to believe that other psychiatric conditions and even other neurological and non-CNS conditions may show evidence of changes in speech latency and its derivatives. So validation is imminent for those of you who really enjoy things that are validated.
I think there's a lot we can do in terms of improving the precision of these vocal biomarkers so that we can better interpret any individual given their age, their culture, their language, given the context, which interview it is, who the interviewer is, much more. And I think, we're getting involved in that process so we can, sort of optimize these vocal biomarkers in a much better way, knowing, for example, when in the session to look at, when people may be particularly struggling with their engagement, and being able to interpret their data for clinical decision making, I think, can be much improved. So, what I envision very soon, hopefully, is rather than a one size fits all cutoff, which we used in this study, a more nuanced interpretation of a person's speech based on their kind of demographic factors and one that can be used to aid clinical decision making.
So, if there's one key message you'd want clinicians and researchers to take away from your work, what would it be?
For me, it would be that very simple speech measures that have been used for decades can easily be measured and used to assist clinical decision making.
I think the key takeaways here are that these methods are no longer shiny toys or curiosities. They're real, and I think there's an opportunity to use them for something that really matters. I think one of the things that has held back the field of digital biomarkers is, in addition to their dizzying complexity, these are very complicated measures in some cases, but also a lack of purpose. It's easy to throw in some cool sounding or cool seeming tools without really understanding what they're for, how they work, what their purpose is. We're not going to see this advance. You know, complexity is unavoidable sometimes, right? There are simply very important but very complex measures that we need to employ. That said, the more simplicity we can finally introduce into our daily work, I think the better off we're all going to be. I'm excited for lots of reasons about this, but most of all, I'm excited to see what comes next. Only other thing I would say, I would like to thank all of the folks who have collaborated with us, who have contributed to all of the work that's been done. Certainly all of my colleagues at Clario, who worked very hard to do sometimes very unglamorous things like organizing audio files and cleaning data. All of that contributes mightily to these advances, and I thank very sincerely all of the folks at Reviva and other companies who put their faith in us. I hope that we have something really brilliant to show for it in the near term.
That's all for today's episode. Thank you to Dr. Alex Cohen and Dr. Mark Opler for joining us to share their work on using speech biomarkers to improve clinical trials in schizophrenia. For our listeners, you can find the full paper in Biological Psychiatry, and we'll include the link in our episode description. To learn more about our journals, please visit biologicalpsychiatry.com. You can find the Biological Psychiatry Podcast wherever you get your podcasts. Subscribe now so you don't miss our next episode, and follow us on our social media channels for highlights and updates. Thanks for listening. This podcast is for informational purposes only and does not constitute medical or professional advice. The views expressed are those of the participants and do not necessarily reflect those of the Society of Biological Psychiatry, its family of journals, or its editors. All rights reserved, including those for text and data mining or use in AI systems.